Skeletal muscle fibers. Origin: skeletal muscle arise from mesoderm, it is mesodermal in origin, except muscle of the iris and myoepithelial cells are ectodermal in origin. According to the morphology, it is classified into striated and non-striated, and according to function, classified into voluntary and involuntary. Types of muscles: skeletal muscle, which is striated and voluntary; cardiac muscle, striated and involuntary; smooth muscle, it is non-striated and involuntary. Origin: prenatal life. During prenatal life, it is arise from cell called myoplast. Cell called myoplast give rise to skeletal muscle fibers. Postnatally, it is arise from satellite cells. Satellite cells, which is flattened cell present around skeletal muscle fibers between the cell membrane and the basement membrane. And the satellite cells are responsible for regeneration and repair of skeletal muscle. As we can see, this is a skeletal muscle fiber, and this is another skeletal muscle fiber. Skeletal muscle fiber, also called skeletal muscle cell, it is acidophilic, and we can see that there is transverse striations. Skeletal muscle fibers are multinucleated. The nucleus is present peripherally under the cell membrane, and it is flattened nucleus. We can see that the nucleus is flattened and peripherally located. So the microscopic characters of skeletal muscle: it is formed of muscle cell, also called muscle fiber. The cytoplasm is called sarcoplasm. The sarcoplasm contains myofibrils and smooth endoplasmic reticulum, which is called sarcoplasmic reticulum. Also, the cytoplasm contains many mitochondria, myoglobin. The plasma membrane of the skeletal muscle cell is called sarcolemma. The connective tissue sheath of skeletal muscle is formed of epimysium, which is dense connective tissue, covers the whole muscle. Perimysium, dense connective tissue also, but covers each muscle bundle. The whole muscle is formed of bundles, and each bundle is surrounded by perimysium. The endomysium is loose connective tissue, surrounds individual muscle fibers. This connective tissue contains blood vessels and lymphatic and nerves. The connective tissue around the whole muscle is called epimysium, and the whole muscle is formed of bundle, and this bundle, each bundle is covered by a connective tissue called perimysium, and this skeletal muscle bundle is formed of muscle fiber, and each individual muscle fiber is covered by a connective tissue sheath called endomysium. And as we can see, the skeletal muscle fiber contains many myofibrils. Cytoplasm is deeply acidophilic, shows distinct striation. Cytoplasm also contains myoglobin and glycogen. Skeletal muscle fiber has multiple nuclei which appear rod-like or ovoid. They are peripherally situated. As we can see, the nuclei are peripherally situated under the sarcolemma. Transverse section of skeletal muscle fiber. When we take a transverse section of skeletal muscle fiber, we can see it is rounded or polygonal. This is a skeletal muscle fiber which appear in transverse section rounded or polygonal. The nuclei appear at the periphery of skeletal muscle, and we can see in the cytoplasm also Cohenheim's areas, which is dark acidophilic dots represent the myofibril in the sarcoplasm. Muscle fiber by electron microscope. As we can see, this is myofibril. This is one myofibril, and this is another myofibril. Each myofibril is formed of myofilaments. We can see also the sarcolemma and the transverse tubules. This transverse tubule represent invagination of the cell membrane into the muscle fiber. Also the sarcoplasmic reticulum, which is a smooth endoplasmic reticulum. We can see mitochondria, Golgi, ribosomes, inclusions like myoglobin and glycogen. The transverse striation seen in skeletal muscle fiber is due to the arrangement of myofilaments in the myofibril. This myofilaments arrangement leads to the formation of a dark band called the A band and a light band called the I band. The I band is bisected by a dark line called the Z line, and the dark band is bisected by a pale area called the H zone. As we can see, this dark band, the A band, is bisected by a pale area called the H zone. This H zone is bisected by a dense line called the M line, and also the I band, the light band, is bisected by a dense line called the Z line. The distance between two Z line is called the sarcomere. The sarcomere is a functional contractile unit of skeletal muscle. As we can see, this Z line, and this is a thin filament, actin filament. The thin filament, actin filament, is anchored to the Z line by a protein called nebulin, and also the myosin filament anchored to the Z line by a protein called titin, and the Z line is supported by a protein called alpha actinin. The thin filament, the actin filament, is 7 nanometer in diameter. It is formed of 3 structures. The F-actin, double helix F-actin, which is formed of a globular G protein, and the tropomyosin, this tropomyosin thread wrapped around the F-actin, and the troponin protein, which is formed of 3 structures. The thick filament myosin, it is 15 nanometer in diameter, and it is present only in the A band. It is formed of double-headed golf club-like shaped. As we can see, the myosin molecule formed of 2 heads. It is a double-headed form and a tail, like a golf club, and it has ATP binding site and actin binding site in the head. It has 2 sites, ATP binding site and actin binding site in its head.